Hi, everyone. I'm Dr. Kelly Blevins. I'm a postdoctoral researcher in paleogenomics at the Archaeology Department at Durham University. And today I want to introduce you broadly to ancient DNA, how we generate data, how we analyze those data, and how we specifically target pathogen ancient DNA. So the first thing you should know about finding pathogen ancient DNA is that it is difficult, very difficult. Duchenne and colleagues provide an estimate of success rate in their recent-ish review paper. And although my percent calculation, which is illustrated in the pie chart there, is a little bit higher than what they suggest in the text, it's not clear how they got 0.01% from those two numbers that they provide. But it's definitely clear that there's a very low successful recovery rate for pathogen genomes, much lower than the recovery rate for the host DNA. And because we all know that archaeological samples are precious and finite, we need to develop more mindful sampling strategies. And this is particularly important for pathogens that are not homogeneously distributed in the bloodstream, because those sorts of pathogens, so those that cause sepsis, those that are coursing through the bloodstream at the time of death, are easily more readily recovered from the dental pulp chamber of teeth. And therefore, they're often recovered opportunistically alongside the targeted host DNA. But for localized pathogens, that's not the case. And so in the next few slides, I'll use Mycobacterium tuberculosis as an example. So that's the causative agent of tuberculosis. And what we know about TB is that it is not distributed in an infected body homogenously. So you can see an illustration of this differential preservation, this differential locality presented here. So for example, at the time of death, the TB causing pathogen could be in the skeletal system, it could be in the lungs, it could be in the abdomen, wherever that localized infection is occurring. And so at the time of death, that's where the pathogen may be, but then as degradation goes on, the chances of recovering that pathogen there are going to become less and less as the DNA breaks down through time. So not only do we have this variability within regions of the body at the time of death, so if TB was in the lungs, for example, it doesn't mean that the bacteria was present on every square centimeter of the lungs at the time of death. So you have this intra-element variation, and it's the same for skeletal elements. Just because there was a bone lesion doesn't mean that the TB DNA, the TB causing pathogen was present in every aspect of that bone. And so there's that, but then that becomes even more and more distorted as decades, centuries, millennia of degradation is occurring. And again, with Mycobacterium tuberculosis as an example, when we're thinking about localized infections, we need to be thinking about the skeletal indicators that an infection was there and the pathogen was present at the time of death. And the clearest way to identify that is through pathological lesions. They act as a flag, they act as a signpost saying, here, there was an infection, I was infected here. And that tells us that the pathogen, the disease-causing pathogen is more likely to be there, and that's where we should sample. And so for TB, you can see the skeletal changes that occur here in these radiographs. So in the first two radiographs, you can see collapsed vertebrae. And that's where the Mycobacterium tuberculosis bacteria will often migrate to in skeletal infections, is blood-rich regions of the vertebrae, the spinal column. They're also more commonly observed in the blood-rich epiphyses or the ends of the long bones in children, which you can see in the radiograph on the right. So these are the types of lesions that translate directly to the archaeological record. We can see them in skeletons ancient skeletons, and we can target them when we're looking for pathogen genomes. But of course, there's still a low success rate, even if we are doing everything we can to look in the right place at the right time. And the failure to recover MTBC DNA can be divided into 4 categories. So the first category and the second category is that there was no MTBC preserved and there is no MTBC or Mycobacterium tuberculosis DNA present. And so we cannot differentiate those two. They will always look the same to us. It's not preserved or it was never there. The third category is that we sampled and TB DNA, Mycobacterium tuberculosis DNA, was there in the element, but we didn't sample in the right place because of, again, this just heterogeneity in the distribution of the DNA at the time of death. And then the fourth category, the best category, is that the TB DNA was there and we sampled and recovered it. So it's a swing and a hit. And of course, this is the goal, this is the dream. So these are the things we should be thinking about going forward because there's been relatively very little work done on the recovery of pathogen genomes and skeletal and the manifestation of skeletal lesions that they cause. And this is something we should be paying attention to because, of course, every time we sample for DNA, we're destroying a non-renewable resource. So this is something we should take into account when we're designing large-scale sampling projects. And in addition to this smart sampling strategy, this paleopathological informed sampling strategy, there are also a couple other justifications we need to make when we're thinking about targeting and recovering ancient pathogen DNA. So the first one is contextual evidence. When that means before you start sampling remains to recover pathogens, you need to ask yourself if the pathogen of interest is likely to be found in this population, in the sample, given the population density, the subsistence strategy, the geography, ecology, climate, the time period, or the social organization. And then you need to ask, of course, about the paleopathological evidence. Based on paleopathological standards, are there index or likely cases at the site? Only applicable, of course, to diseases, pathogens that affect the skeletal remains. So this isn't applicable in every case, every situation, but still it's something you need to consider. And then, of course, the execution of a successful pilot study. So before you sample widely, before you remove skeletal elements from their skeletons, their skeleton boxes where they're stored to be shipped off and destroyed in a lab, you need to demonstrate that the pathogen DNA is preserved and it's there to find. So again, yeah, can you demonstrate that the DNA of interest is preserved and that preservation is sufficient for genome recovery? And going back to this informed sampling selection, so you can't get every type of DNA that you want from a single skeletal element. There are certainly skeletal elements that are commonly sampled for DNA, and those would be teeth and petrous portions, which I'm sure you've heard about before. But when you're targeting pathogen DNA, you need to think critically about this because you can only ever recover what was actually there to begin with. So thinking about sampling teeth, and when we sample teeth, we don't sample the enamel, right? We sample what's in the tooth. We sample the organic portion, the pulp chamber, or the dentin inside the crown. But the pulp chamber is the most relevant for recovering pathogen DNA. And that's because pathogens that cause septicemia or this sort of just unregulated wild distribution throughout the bloodstream of the pathogen, this cascading effect of of pathogen distribution throughout the body, septicemia, that can be detected in the pulp chamber. And we think it's because their residual blood is better preserved there than other parts of the body within this pretty well protected pulp chamber inside this, the enamel crown. And so it's a place to target if you're looking for pathogens that cause septicemia, such as Yersinia pestis, or the causative agent of plague. You could also detect oral pathogens here, or pathogens that infect the oral cavity and rhinomaxillary area. So there's also been successful recovery of Mycobacterium leprae, or a causative agent of leprosy from teeth from pulp chambers. Another substrate you could target when you're looking for pathogens is dental calculus. So again, this is similar to the pulp chamber pathogens, in that, again, thinking about what is localized in that area, what are localized infections are likely to be there. And again, it's oral pathogens, and it's pathogens that infect the oral cavity and rhinomaxillary area. So for example, the pathogens responsible for stimulating the inflammatory response of periodontal disease, you would maybe be able to pick up that dysregulation there if you were looking at dental calculus. And then lastly, of course, bones. So when you're sampling bones for pathogens, it doesn't make sense to sample a femoral mid-shaft if you're looking for Mycobacterium tuberculosis DNA, because you have no indicator, no reason to think that the DNA, the pathogen itself, would have been there at the time of death. So when we're sampling from bones, we need to be thinking about where the localized infection was likely to be. That's where the pathogen was most likely to have been at the time of death. And that's where we should sample. So again, looking for lesions that indicate the pathogen was there at the time of death. And in some cases, there's been some success of recovering pathogens that cause septicemia. So again, this widespread distribution of the pathogen throughout the body in skeletal elements. But this is rare, and this is the exception, definitely not the rule. So once you've identified the substrate that that's most likely to contain your pathogen of interest, you then must extract the DNA. And so most ancient DNA extraction methods follow the same workflow that's presented here, and uses both the mechanical and chemical techniques to extract and isolate the DNA. So first, the bone or tooth sample must be pulverized to really increase that surface area that's exposed. And this is done ideally with something minimally destructive like a dental drill, which you can see here. And we try to use 50 milligrams or less of powdered material. The powder is then demineralized and digested, which degrades contaminant macromolecules like proteins, and ideally releases the DNA in solution. Then the solution is forced through a silica membrane where the DNA gets stuck and is bound, and everything else is washed through. And then the final step is releasing that DNA, which will be bound in the silica membrane in a buffer that breaks that attraction with the silica membrane between the silica column and the DNA, breaks that attraction, so the DNA is eluted in your buffer, which you see here, and you have your tubes of DNA extracts. So the DNA extract, if you could look into it and see all the tiny fragments, you would see, well, you wouldn't see because it would all just look like DNA, but trust me, it contains DNA that was all of the DNA that was present in your sample. So it contains the host DNA, it contains the pathogen DNA if it were there. But more than either of those two sources, it contains the contaminant DNA. So the DNA of the archaeologist who excavated it, DNA of the mole or rat that crawled across the skeleton in the burial, the DNA of the museum curator, your DNA possibly, it contains all sources of DNA. But then to amplify or sequence all this DNA and keep all this information, so all of the the host DNA, the pathogen DNA, the microbiome DNA, all the DNA that could be potentially informative from the sample. In order to keep all of that information downstream, we need to build it into a library. And DNA libraries are built by plopping on a DNA sequence with a known composition onto every single DNA fragment in the extract. And so that's what's being illustrated here. So you have this nice little double-stranded double helix, which represents the adapter sequence. That gets attached or ligated to every DNA fragment in your extract, turning it into a library. And so we could say that a DNA library is an organized DNA extract. All of the DNA fragments have been tagged and labeled with a universal DNA sequence that can be targeted during downstream amplification and sequencing. And that's important because in order to sequence the DNA, it needs to have a way to bind or attach to complementary base pairs on what's called the sequencing flow cell. It needs to, you can think of the adapter basically as a little hand that allows it to grab onto the flow cell and be sequenced. So not only do we need these adapters on our fragments in order to make more of a library, to amplify the library, but also for sequencing to generate the data. And DNA libraries can be sequenced directly via shotgun sequencing. And shotgun sequencing is when a random subset of the library is sequenced, which you can see illustrated here. So depending on the preservation of the sample or how much contamination there is, you'll get a variable amount of host DNA and contaminant DNA. And generally, if it's there, a very, very small, relatively small amount of pathogen DNA. For example, here the pathogen DNA is shown in purple. And if the pathogen DNA is preserved, it will be in the library, but you'll have to sequence very, very deeply. And that means sequencing deeply means sequencing more. So billions, billions, billions of reads, maybe trillions of reads to get sufficient amount of the pathogen DNA to reconstruct a genome. And as it probably sounds, it's expensive to sequence trillions of reads, and it produces a truly unruly amount of data. So that's why we often employ something called targeted enrichment or targeted capture. And it's very useful for overcoming these problems of small relative starting amount of our DNA of interest. So as I said, pathogen DNA constitutes proportionally very little of the sample. So we need to ramp up this relative frequency of pathogen DNA fragments. And target capture entails designing baits or making baits from chopped up genomic DNA of a closely related organism or the organism of interest and mixing those baits with the sample library. So that's what's happening here. You have made your baits complementary to Mycobacterium tuberculosis DNA. You have manipulated the temperature so that all of your double-stranded fragments have disassociated from each other, and they're all floating around single-stranded so that these baits can reach your Mycobacterium tuberculosis DNA. You manipulate the temperature some more, and these baits will hybridize or bind to your targeted DNA. There are magnetic beads attached to these baits, and then you physically remove your bait bound fragments from all the other fragments in the solution with a magnet. It is pretty awesome to watch, and it's pretty much magic. But after a round of PCR, you eventually ideally have only your fragments of interest left in the solution. Of course, it's never actually 100% because non-target DNA will tag along for the ride, but this is what you're shooting for. So sequencing the capture products. So when you sequence a subset of your enriched library, assuming it actually had enough pathogen DNA to recover, you recover proportionally much, much more pathogen of interest, which again, you can see here with purple. So to summarize, target capture is the efficient way to sequence your organism of interest from a DNA library. And it's the most efficient way to pull out the DNA you're interested in from a sample in which it may constitute much less than 1%, 0.01%, 0.001% of the whole. So once you've enriched a library for pathogen DNA and sequenced it, you get back tons of A's, C's, T's, and G's. So your DNA bases, adenine, cytosine, thymine, guanine. Once you've done some preliminary tidying up of the fragments, the first major step of the analysis is to map them to a reference genome to see if you actually have enough reads to make up an entire genome. And the quiet literally looks like this when it's visualized. So each purple line here represents a DNA fragment that was sequenced into a read. And there are some key terms that go along with thinking about your successful recovery of your target organism that I want to introduce you to and go over. And the first one is coverage. So we would say that this genome, which it's just a toy example, right? So we're calling this short reference sequence here the genome. These are our sequenced reads from our DNA, our enriched library. And we would say here that this library has 100% coverage. This sample has a 100% coverage of the reference sequence. So that means that all bases in the reference sequence, which you can see here, are covered by at least one unique read, one unique sequencing read. Likewise, this sample has approximately 75% of the reference genome covered. You can see, of course, this big gap here. So only about 75% of the bases in the reference sequence are covered by a unique sequenced read. Another key term is coverage depth. And the difference here is, instead of looking at just if each reference base is covered by a read, this is a summary statistic of how much each base in the reference sequence is covered. So for example, this cytosine here, the C, we would say that it has a 10x depth coverage because it is covered by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 reads. And we would say this adenine here, it has 11x depth of coverage because it is covered by 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 unique reads. And again, same logic, we would say that this adenine here has an 8x depth of coverage. 1, 2, 3, 4, 5, 6, 7, 8. And we can average that across every position in the reference genome to get what we would call an average depth of coverage. A lot of papers report this as, they will just call it average coverage, or they will call it coverage, but they mean average depth of coverage. So don't get confused there. And this is the most commonly reported metric in ancient DNA papers, this average depth of coverage. And for this toy example here, we would call it a 9x genome, a 9x average depth of coverage. And what we're saying is that on average, every base in the reference sequence is covered by 9 unique reads on average. And this is the ideal scenario here. So we have 100% of the reference genome covered and with greater than 5-fold depth of coverage. But unfortunately, this is what we have to work with generally when analyzing ancient DNA. And it's a patchy percent coverage and a high standard deviation of coverage depth. But even when our data look like this, we can still answer some pretty cool questions. And so that brings me to my last slide, which is how do we answer questions using ancient pathogen genomic data. And to think about that, we need to think about evolution and the way genomes behave, and phylogenetics. If you have ever read an ancient pathogen DNA paper, you all have seen a phylogeny. Well, a more modern ancient pathogen DNA paper where they recovered the whole genome, or at least some sequences from the pathogen, you will have encountered a phylogeny. And so I just want to tie, genomic evolution, and phylogenetics together, so you have some sort of toolkit when you're reading these papers for the first time, or encountering these papers for the first time. So to make it really simple, to think about what's actually happening, if you look at all of these bacteria here, each one of these cells has a genome, right? Or a chromosome. And that's just one, we can think about it here in this toy example as this linear stretch of DNA, A, C, T's, and G's. And mutation creates variation in each of these genomes. That's illustrated here by these red bases as opposed to the black bases. So in these two bacteria, in these two bacilli, they have a G at this position, where in this bacterium, there's a C, right? So there's variation within this colony of bacteria. And this bacterium has an A here, right? Instead of a T, opposed to the other two. And so by reproductive success, by chance, these mutations can be fixed or they can be lost completely as the life cycle of the bacteria continues. And so what you're seeing here is that this A has been fixed in a population. And so over generations, you can imagine that these, we'll call them variants here. These variants are going to accumulate in different ways in different lineages of bacteria. And so when we're trying to understand how pathogens were distributed in the past, how ancient pathogens are related to other ancient pathogens, or how all ancient pathogens are related to pathogens that we encounter today, modern pathogens, the first thing we want to know is how they're related to each other. And this is one of the most common ways to do that. So we can basically take these genomes with these variants, these mutations in them, and we can organize them based on the mutations that they do or do not share. And this is going to illustrate their relationships. And that's what you're seeing here in this tree-like format, in this phylogenetic tree. So, for example, what this is illustrating for us, if we look at this line here, what we're seeing is the unique evolution of individual C, of bacterium C. And so this line represents all of the mutations that are unique, all the variants that are unique to C that are not shared by these other bacteria. And the same, of course, is true here for bacterium D. So this is illustrating the unique evolution of bacterium D, all the variants that are unique to D right here. And what these internal branches are illustrating is the shared evolution between these terminal ends. So right here, what you're looking at is a representation of all of the shared variants between bacterium C and bacterium D. And so this is how we visualize and make inferences about ancient pathogens and their relationship to modern pathogens today, how they've changed through time, how they're related to each other, how they're related to how and if they're related to modern lineages. So that was a very fast crash course into ancient DNA, paleogenomics, and ancient pathogen DNA, but I hope it's given you some useful tools for thinking about this research, maybe planning your own research and navigating the literature. Thanks for listening.